We begin with the scatterplot view. The scatterplot view visually represents cells from various species. In this view, each point represents a cell arranged by its low-dimensional embedding, with colors indicating predefined cell types. This helps us see the distribution of different cell types across the datasets. Next, we move to the hierarchy view. In the hierarchy view, we see a three-level hierarchy of cell types. At the top, we have a class-level category like neuronal and non-neuronal cells. Below that, the subclass and cluster levels reveal a more detailed classification of the data. The scatter plot is colored according to the lower level of the hierarchy view, which is the cluster level. In the system, exploration typically starts by selecting a cluster of interest. For this demonstration, let's select the chandelier cluster by clicking on it in the hierarchy view. The selected cells are instantly highlighted in the scatter plot. This helps compare the selected cells with the broader dataset. Once a cluster is selected, the next step is to explore the abundance of these cells across the species. Further, it is also interesting to identify the most differentially expressed genes in the selected cells. We compute this information, which might take a moment depending on the size of the selection. When the computation is complete, we shift our focus to the table view. In this view, we can see both the relative and absolute abundances of the selected cells across species. On the left side of the table view, we see a list of genes that are highly differentially expressed in the selected cells. The appearance column indicates how many species have that gene among the top 10 differentially expressed genes for the selected cell type. For example, since the ERB4 gene is listed as the top gene for 7 species in our selected cell type, it means ERB4 is one of the interesting genes for this cell type. To dive deeper, we can explore these genes further to discover if certain genes have potential cell type specific marker features. Therefore, we try to find genes that are highly expressed for the selected cells compared to the rest of the cell types in the dataset. By clicking on a gene, we update the 3 different views. In the table view, we see the mean differential expression values within species. These highlight how prominently this gene is expressed in the selected cell compared to the other cells. The ranking column shows the gene's position for each species based on its high differential expression values. The genes with higher expression will have a lower rank. In the table, the species which have a rank within the threshold is selected as interesting species for further exploration. These generally have high differential expression values compared to the rest. The scatterplot view also updates to show all the cells in the dataset for the selected species. The points are colored by their gene expression values. This allows us to visually compare the expression levels of the cells to other cell types in the dataset. This helps us determine if the selected cells have higher expression levels for that gene across the selected species. Additionally, by clicking on rows in the table view, we can explore the expression values one species at a time. This reveals how gene expression levels shift across different species based on ranks in the table, becoming from more distinct to uniform. The next step in our exploration is the phylogenetic tree view. In this view, we can map and observe expression patterns across evolutionary branches, linking findings to the phylogenetic hierarchy. For example, branches containing pig, cat, coyote, and ferret in the tree view show high mean expression values, whereas the great apes branch has low expression values with gorilla as a notable outlier. Through this exploration workflow, we can compare expression patterns across both individual species and broader evolutionary contexts. By selecting different branches or species, we can gain further insights into how these patterns vary. Additionally, we can explore other attributes like relative cell abundances within the phylogenetic tree. For example, higher cell abundance can be seen in the upper part of the tree compared to the lower part, highlighting patterns that might not be apparent in other views.